Eric did his undergraduate and his PhD at Oklahoma State University. And we started working with Eric when we had a joint project with funded by the Department of Defense while Peggy and Liz and I were at Indiana University, and Eric was on that project. It was totally awesome to work with. So we jumped at the chance to recruit him when he finished his PhD with Gail Lipton to come here, and he's going to talk about two of the projects that he's worked on while he's here. And one of them was funded by the Max, and the other one is funded by Eric's grant with the U.S. Department of Agriculture. Eric?
All right, thank you, Jim. Okay, so as Jim said, my name is Eric Duell. If we haven't had the chance to meet, I started here at KBS-CER in January of 2022, and today I'll be trying to merge two similar but very different projects that I've been working on since I got here. And most of my research in the Bever Schultz Lab focuses on the interactions between plants and their associated soil microorganisms, and how those interactions facilitate proxies for or ecosystem services themselves. And so without further delay, we'll go ahead and perfect. Okay, so just a little bit of an outline. This first experiment I'll be talking about was a pretty large, complicated greenhouse experiment that Dr. Tom McKenna, Liz Kojal, Jim, and Terry Lucky helped out a lot with, looking at the effects of plant assemblage, soil microbiome, and related land use history effects on soil stability and nutrient leaching through the soil profile. So we'll dig into the methods and results of that a little bit, and then we'll move on to this larger long-term field study that looks at some similar measurements out in the field over obviously a longer period of time, using plots at the KU field station that were established back in 2018. So most people in this room are probably very familiar with grasslands and the importance of grasslands, but I do like to start off most of my talks about grassland ecology in a global context. So grasslands encompass about 40% of Earth's terrestrial ice-free surfaces on 6 of the 7 continents, every continent except Antarctica. And they also coincide with about 80% of Earth's agriculturally productive land, which, as you can imagine, causes a lot of tension and conflict between agriculture and conservation. Again, most people in this room are probably very well aware of that. And while as ecologists and conservationists, we don't always like to put things into terms of their benefits for us, but that's one easy way to convey the importance of these systems to the general public. We talk about ecosystem goods and services. So goods being those products that we can derive from an ecosystem that benefit us in some way, so food and fiber. And then ecosystem services, those usually biogeochemical processes or those physical processes by which life on Earth is sustained. Some of the big ones, some of the key ones, but by no means an exhaustive list. In grasslands would be aquifer recharge and groundwater recharge, carbon storage or sequestration, nutrient cycling, also pollination services. While they are called grasslands, most healthy grasslands consist of a large component of forb species or broadleaves, which sustain pollinators. About 60% of which are crop species globally depend on some sort of pollination by either invertebrates or birds or bats or something like that. And then other ecosystem services like soil stability, which obviously prevents erosion. So grasslands provide a whole suite, again, not an exhaustive list, but a whole suite of ecosystem goods and services that really highlight their importance. But along with being the most widespread of Earth's terrestrial biomes, grasslands are also widely considered to be among the, if not the most imperiled terrestrial ecosystem, and that's due to a number of co-occurring and interacting forces, primarily conversion to robotic agriculture. That's especially prevalent here in the Midwest of the United States, but also other co-occurring forces like climate change and drought, which facilitate desertification. So on the peripheries of most major deserts in the world are grasslands, just due to those similar climates. And most of those grasslands on the periphery of large deserts are at risk of desertification or spreading of those dune systems. We also have invasive species. That's especially problematic here in the tallgrass prairie and in the Flint Hills. So even in areas where land wasn't plowed because of that shallow bedrock, invasive species and woody encroachment are further threatening these imperiled systems. And a lot of that due to fire suppression and encroachment of native woody species, and in some parts of the globe, also urban sprawl. And again, the effects of the extent of these grassland losses again are especially evident here in North America. I know you can't see that very well, but by most estimates, anywhere from 1 to 10% of the historic extent of the tallgrass prairie in Central North America remains, and that's referring to pre-European settlement. Other parts of the globe, while this is a worldwide phenomenon, this loss of grasslands, other parts of the globe are faring a bit better, with parts of Brazil, the Cerrado region at about 20%, and then Africa, Asia, and Australia between 50 and 70% of those grasslands remain. So much better off than the US, but still a global phenomenon worldwide. And these land use history impacts have long-term, sometimes decadal or even century-long impacts that are really, really difficult to restore. This happens at both the population and community level, scaling all the way up to the ecosystem level, where we see reductions in biodiversity and especially below ground. And this reduction in biodiversity and the simplification of these soil microbial communities can lead to soil erosion, loss of those soil aggregates, which are those important ecosystem services or proxies for ecosystem services that that are imperative to grassland health and the health of diversity and humans as well as nutrient leaching being problematic. So, and that brings me to kind of the focal organisms of my research, the plants and especially the soil microbes. So soil microbes fill, as you guys are all very aware, a variety of trophic levels and perform a variety of roles in ecosystems. They act as decomposers, facilitating the breaking down of organic material and the recycling of that within the system. They also act as pathogens and mutualists, which facilitate and impact plant community structure and function, with pathogens doing things like limiting the dominance of a single species and lots of in native intact systems. And soil microbes also sometimes act as canaries in the coal mine, for lack of better terms, as sentinel species for ecosystem health. When you start to see these skewed fungal to bacterial ratios and things like that, it's a sign that something might be up and that ecosystem is being impacted. And you might see that before impacts to other larger organisms, more easy to see organisms. So taking that one step further, and if any of you if you guys have been at KBS for any amount of time and seen any of Jim or Peggy's students talk, you've probably heard them talk about arbuscular mycorrhizal fungi, and I don't want to bore you with that, but they are really cool, really important, and that's the focus of a lot of my work. And just real quick, if you're not familiar with the symbiosis, these microscopic fungi live in and around the root systems of plants, exploring and mining rhizosphere soil, uptaking critical essential elements or nutrients like phosphorus and nitrogen, as well as soil moisture, shuttling that back to the plant, penetrating the root cortex. And in exchange, the plant gives anywhere from 10 to almost a quarter of the carbohydrates it produces via photosynthesis to the fungi itself, which I think in and of itself, those numbers are a bit of a testament to the importance of this relationship. In fact, many hypotheses suggest that arbuscular mycorrhizal fungi facilitated the colonization of land over 400 million years ago by vascular plants. So this is an ancient mutualism that's prevalent across the globe in almost 80% of terrestrial vascular plants. So very important and very pervasive in the plant world. And arbuscular mycorrhizal fungi are also pretty well correlated with several ecosystem services. Some earlier work by my PhD advisor Gail at Konza Prairie over in Manhattan, Kansas, suggests a high correlation between mycorrhizal hyphae in the soil and a glycoprotein they produce, glomalin, which helps bind soil particles and promotes the aggregation of soil particles, which then facilitates increased carbon storage capabilities of those soils. So mycorrhizal fungi, again, linked to several ecosystem services, namely aggregate stability and further down carbon. So the big question that sort of unifies these two, again, similar but very different experiments, is how do AM fungi and other soil microbes interact with plants to influence these soil-related ecosystem services, as well as above ground community productivity. But specifically, we're looking at things like soil stability, soil aggregates, and carbon stocks. So first, I'll be talking about the greenhouse experiment. The goal of this experiment was to assess and elucidate the effects of the identity of the plant community, or the plant assemblage, as you'll see here in a little bit, the land use history, and the associated microbiomes of these to these soil-related ecosystem services, or proxies for ecosystem services, and then determine the degree to which these are driven by these plant-soil interactions or plant-soil processes. So it was a pretty large experiment, and the design is a little complicated, so I'll try to walk you through it as simply and efficiently as possible. So we started out on this top left-hand side with 4 plant communities. I put that in quotes because some people do take issue of calling it a community when there were only 3 plants in a microcosm or mesocosm study. So I interchange communities or assemblages, but these were either annual ag, perennial ag, post ag, which is kind of that go back land where historically it would have been pasture cropland, and then allowed to return to, you would hope it would return to a native state, but typically it's dominated by non-native species that were planted there for pasture land. And then late successional native prairie plants. So in combination with this, we had 5 soil inoculum too. So sterile soil, AM fungal isolates, so those AM fungal propagules, either spores or hyphal fragments that were introduced, so that removes the effects of the rest of the soil microbiome. However, these final 3, annual ag, post ag, and remnant native prairie, those are whole soil inoculum, meaning they contain pathogens, fungi, bacteria, nematodes, any microbe that would have been in that soil. And this was replicated across 3 sites as well. So Jefferson County, up where the field station was, Anderson County down at Welda Prairie and near Ottawa, and Konza Prairie Biological Station over outside of Manhattan, Kansas. So with all those in mind, you can see this turned into a fairly large experiment fairly quickly. And for the botanists and plant ecologists that are in the room that are interested in the identity of the plants inside these pots, each community or assemblage was represented by a single grass species, a single legume species, and a single aster species. Those three taxonomic families are the most well represented families in prairie ecosystems and many ecosystems across the globe. So that's a common theme throughout the rest of my talk. So corn, soybean, and annual sunflower as our annual ag plants. Our perennial ag plants were Kernza, alfalfa, and prairie rosinweed, all of which are used by the Land Institute in attempts to perennialize agriculture. Post ag, which would be these non-native invasive species, smooth brome, yellow sweet clover, and unfortunately, I did have to use a native annual prairie species in these go back or old field or post ag lands. I said they're typically dominated by non-native species. If there are native species there, they're typically the early successional ruderal species, and none of my perennial post-ag weed species germinated, which is surprising. So I was left with a decision to make and found some annual native plants to use. And then our native prairie plants, little blue stem lead plant and prairie blazing star. And just to give a close-up of what these looked like just a couple weeks after planting, and this shows these pictures were all taken at the same time, and so these plants are all about the same age, and you can see on the far right-hand side just how below growing these native late successional species are compared to the other species used in our experiment, which really comes into play when we look at the results here in a little bit. And so the inocula came from annual ag field, post ag field. or remnant native prairie. That's a picture of Congo. And so this is where we were testing soil detachment, which is also known as erosion risk, which is that proxy for soil stability. That's me looking like I'm having a very good time. It was pretty fun. Tom McKenna really helped design this flume. He was the mastermind behind this. Using several other studies in the literature, we used the materials to build a flume system. And so at the end of the experiment, we used a hammer core or a slide hammer attached to soil core with a plastic sleeve insert that pulled the soil core from the center of the pot. And then after we pulled those soil cores, we were then able to insert that underneath this gut ring, this flume, and we had a hydraulic pump underneath it that slowly pushed the soil core up to maintain a level just above flush with the flume itself, running a consistent flow of water, a consistent volume and speed of water over the soil, and then catching these detached soil particles, drying it down and weighing that to get an estimate of soil loss or soil detention. And this is several different angles of this contraction and the modified flume design from these papers. We allowed 60 seconds of simulated overland flow unless all of the soil was washed out of the core, which did happen on a few occasions because some of these samples had very little root, very little soil structure. And that was mostly in some of the annual ag pots, as you would expect. We also, with the help of Dr. Lucky and his lab, attached these ion exchange resin beads to catch phosphate and nitrate ions that were leaching through the soil profile to get an estimate of nutrient leaching in response to the interaction between, again, soil microbiome, land use history, and the plant assemblage there. These two were targeted because they are the major contaminants in water in agricultural systems. So just to rehash those measurements we took: plant productivity above and below ground, soil detachment. We sent samples off for aggregate stability at the University of Missouri, where they give you back a proportion of water stable macro aggregates. So those aggregates that aren't breaking down when exposed to water or flow, microbial abundances, and nutrient leaching. So jumping straight into the results, this is on a log scale above ground biomass. As you can see, most of the bars are pretty similar to one another. So not a lot of difference in biomass production, although I do want to highlight the native plants and how they perform in sterile soil. Again, just very convincing that our native plants are surprisingly, not surprisingly, very dependent on fungi. And that's something that's very well established in the literature. And I guess just to explain this quick, along the x-axis is the plant assemblage itself, and along the y-axis is that biomass, and the colors represent that soil inoculum. So now moving on to look at the relationship between below ground biomass and soil detachment. Along the x-axis, plant assemblage again, along the y-axis, root biomass. So the ag plants didn't produce much below ground biomass at all. That's not surprising because these plants were bred to produce a lot of above ground biomass very quickly. And almost perfectly inverse of this is the soil loss with plant assemblage, where the annual ag plants lost the most soil. The post-ag plants, because they're typically non-native invasive species, they have these characteristics that allow them to really infiltrate and invade ecosystems, one of those being high growth rates. And apparently that is happening below ground too, just producing a lot of root biomass. And those post-ag plants also actually facilitated facilitated soil stability. And again, want to highlight the native prairie plants performing very poorly in the absence of a living soil microbiome, and that resulting in pretty substantial soil losses. So just to show some simple correlations that really drive home the point of that relationship between soil loss and root biomass, with soil loss increasing as root biomass decreases. soil detachment and the water stable macroaggregates. So now along the x-axis, we have the proportion of water stable macroaggregates in a soil sample, which as that proportion increases, soil loss decreases. So you're losing less soil. And then looking just real quickly at some of the nutrient dynamics, a living microbiome promoted nitrogen retention. So the greatest nitrate loss was in the absence of a living microbiome. And then looking at the plant assemblages, the nitrate losses were actually lowest in plants, which makes sense because these plants are growing really big, really fast, and are nitrogen hungry, as opposed to on the far right, the native plants they're small, they're still small at this point, they're slow growing, and they're not as nitrogen starved or nitrogen hungry. So in conclusion for the greenhouse experiment, the below ground biomass and soil macroaggregates were strongly linked to soil detachment, which is what we would expect. We saw overall little evidence of soil inoculation effects, especially for the native plants, which we would have expected. We did see, sorry, I misspoke there. Strong microbiome effects on the native plants, little effects overall. And then we did see that the microbiome was strongly linked to nutrient retention, although it was just the presence of a living microbiome. And then I guess one large caveat to this study that some reasoning maybe why we didn't see the results that we would have expected was that this was conducted over a single growing season, and these native plants are long-lived perennial species that likely require more than a single season in the greenhouse to establish a large, robust root system and produce a lot of above ground biomass. So with that, we'll move right into the field experiment. Okay, so the goals and objectives of this study were to assess the plant community richness and phylogenetic dispersion effects on community productivity, to assess the role of the soil microbiome in the facilitation of this productivity biodiversity relationship that's so frequently seen in ecosystems, and then examine the relationships between the plant and soil microbial community diversity and how these relate to some of those below ground ecosystem processes that I've mentioned in previous slides. So this experiment started out with a pool of just starting very coarse, started with a pool of 17 plant species. And if this sounds familiar, maybe you've heard Laura Podzakowski talk. She worked on a very similar experiment using the rainout shelters. My research was not conducted under these shelters, but in an adjacent similar experiment. But we started out with a pool of 17 species from those same 3 taxonomic families that I mentioned for the greenhouse study. Asteraceae, Fabaceae, and Poaceae. The plant species richness in these plots was manipulated to levels of 1, 2, 3, 6, or 17 species. You see in parentheses there, it says or 5. That's because after 2 years, one species of grass failed to establish, so that after 2 years, that was removed from experimental design and analyses. So initially, that would have been an 18 species mixture as our highest diversity, but that was reduced to 17 species after that failure to establish. All of these species were equally represented in both monoculture and mixture, and phylogenetic dispersion was also manipulated to where the 2, 3, and 6 species plots were either phylogenetically underdispersed, meaning represented by a single taxonomic family, or overdispersed, meaning multiple taxonomic families. And obviously the single species would be from a single family, and 17 would always be from multiple families. So this experiment also manipulated the soil microbiome, where we had 3 soil inoculation treatments: non-inoculated or sterile soil, whole prairie soil, and AM fungal isolates. Again, that isolates just the effects of the AM fungi themselves. It is worth noting that both the non-inoculated soil and the whole prairie soil, that soil about the top 2 inches of that soil was raked in from Weld Prairie down in Anderson County. There was a roadway expansion project going on. So literally tons of soil were brought up to the field station and raked into those two plots. But the non-inoculated or that sterile soil was steam pasteurized first before being raked in. And the AM fungal isolates, no soil was added to those. This was historically a brome or pesque field, and that soil was just simply tilled up and then inoculated with AM fungi. These seedlings were all started in the greenhouse, and the complete experimental design consists of 432 2¼ meter squared plots. So a very large experiment out there. And unfortunately, we don't have any great aerial imagery at the moment of this experiment, so I'm gonna try to do my best with these PowerPoint designs to explain what is going on here. So this is a randomized block design consisting of 2 blocks and 3 sub-blocks within each block. Each color represents a soil inoculation treatment, which I'll refer to as a treatment, with the red or the salmon color being that whole prairie soil, the purple being the AM fungal isolates, and the white being the sterile control. And let's see, so the treatments were not completely randomized within each block because we had to deal with the movement of the inoculum itself between the plots. So similar treatments were grouped together in groups of 24 plots. So you can see there plots 1 through 24, which I've zoomed in on here, all consisted of the prairie AM fungal treatment, and within each plot consisted of either monoculture, biculture, 3, 6, or that should say 17 species, and then 2 blanks where no seedlings, no plugs were planted. And a total of 18 seedlings were planted into each of these plots, with the exception of those 2 blanks, in this sort of orientation or design. And the number of seedlings for each respective species was going to depend on the species richness treatment of that plot itself. So this plot 1, consisting of Coreopsis and Dahlia, with it being 2 species, would have had 9 Coreopsis individuals and 9 Dahlia individuals. Plot 2 would have consisted of 18 Eupatorium individuals, and then looking at plot 4, each of those plants would have been a different species planted into that plot. So the productivity measures for the last 5 years, about once a month, visual cover estimates were made to get percent cover of each of the species of interest. And also at peak biomass production, biomass was harvested in these 0.1 meter square strips, and where that was then sorted, dried, and sorted by species, dried and weighed, and then scaled up to get plot level estimate productivity for each species and total plot productivity. So one of the purposes of this experiment was to test over yielding, which suggests that species are producing greater biomass in mixture relative to their own productivity in monoculture. And this can occur through 2 primary means, either selection or complementarity, with selection suggesting that the most competitive plant species is producing most of the biomass. So just by chance, you're likely including a highly productive species like big blue stem in these plots, or complementarity when many of the species are producing greater biomass in mixture relative to in monoculture. And with the help of Laura Podzakowski, conducting some additive partitioning methods to tease out these selection and complementarity effects, and then the sum of those being the net ecosystem or net biodiversity effects. Oh, moving on, I just really quickly want to show you the effects of species richness on productivity. So we are following that same general pattern of increasing productivity as species richness increases, regardless of inoculation. However, in most of these biodiversity ecosystem function or biodiversity productivity experiments, complementarity appears to be the mechanism which really drives the long-term productivity benefits. So for this part of my talk, I'll be specifically touching on that And looking along the x-axis at soil inoculation and along the y-axis, that complementarity effect, we really see the impact of that living soil microbiome, that tightly linked co-evolved microbiome in generating complementarity effects. However, when we break that out by plant family, whether it was in a single family plot or in mixture, we start to see some pretty variable results, with it being very dependent on the plant family itself, which of these inoculation treatments is really driving or generating these complementarity effects. Most notably, or most interesting to me, is that in Asteraceae plots, so the furthest left category on that right-hand panel, complementarity seemingly very much generated by the prairie AMF themselves, so not that whole soil microbiome. However, we see the opposite pattern with Fabaceae, or the legumes, where a lot of that complementarity is being generated by the whole prairie microbiome, meaning the pathogens, the nematodes, the bacteria, something else in that suite of microorganisms. And overall, again, in mixture, we see similar patterns as the main effect of inoculation, where the whole prairie soil and the AMF isolates are generating complementarity relative to the uninoculated plots. So that was the complementarity component as it relates to soil inoculation. Now we're moving to those net biodiversity effects, which again is that some complementarity and selection. So first, this is looking across year on the x-axis. So these measurements were taken over the last 5 years, and on the y-axis is that net ecosystem effect. And there's lots of dots, and they're not really see-through, so it's hard to see those patterns. But what I want to point out is the yellow, or this kind of salmon or orange colored line, and the green line. So that's Asteraceae and Fabaceae that are almost completely flat. So really no benefits. So no increase in net ecosystem effects across time in the absence of that soil microbiome. When we look at just the AM fungal isolates, we still see Fabaceae remaining a relatively flat line, so not really generating net ecosystem effects in Fabaceae with only AMF. But then whole prairie soil, the mixture as well as each family by itself, showing a steep increase over time in those net ecosystem effects. So now moving on to some very new data that I've just analyzed, and this is looking at some of those below ground processes in response to these treatments. So looking now at root biomass and species richness. Along the x-axis is species richness, and along the y-axis is root biomass. So we do see a significant steady increase in root biomass as species richness increases. And then along the x-axis, we have that phylogenetic dispersion. Along the y-axis, root biomass again with Poaceae. So that third point, that's the grass family producing a lot of biomass. So, and that makes sense. Grasses make a lot of fibrous root biomass, and a lot of fibrous roots weigh quite a bit more than a handful of tap roots or thicker fleshy roots. We see this pattern mirrored with the water-stable macro aggregates, and that's not to say that that's not being driven by microbial activity. Unfortunately, I don't have the microbial data just yet, still waiting on that, but there's lots of evidence out there that suggests more roots in the soil that's stimulating microbial activity, which would then produce those exudates and those secondary compounds that facilitate aggregate stability and the binding of soil particles. So now moving on to some of the soil nutrient data, total nitrogen. While this wasn't significant, I have that p-value down there. We did see a small increase across species richness treatments in nitrogen, and this was significant, an increase in carbon in the soil as species richness increased. So now looking at a marginally significant interaction between carbon, species richness, and soil inoculation, and focusing in on that. So There were just inherently lower soil carbon levels in the AM fungi, and that's likely because of that dynamic that I mentioned earlier with the whole prairie soil and the non-inoculated or the sterile soil coming from WELDA, and WELDA soil likely having just higher carbon levels initially. But looking at the slope of the line, we do see the AM fungi treatments being a steeper slope and likely facilitating or driving that interaction pattern we're seeing. So that's I know that's a lot of data. In future directions, there's still lots of data forthcoming, particularly regarding the soil microbial community. We've got several other people in the lab that are looking at plant soil feedback dynamics, weed suppression, community change over time, just lots of cool stuff going on out there, and still lots more analyses to do on this data. And I guess overall conclusions, there's several very simple ones, and that's that the plant and soil microbiomes are intricately and intimately linked, and that disruption of these relationships can have consequences all the way up to the ecosystem level, and that these are critical for the restoration of these ecosystems as well. So lots of acknowledgments, particularly KBS and EPSCoR and USDA NIFA that have funded my postdoc thus far, and lots of people in the Bever Schultz Lab, the Ecology Lab. And yeah, with that, I'll take any questions.